Alright, so here we have some DNA. On the strand we have a PAM site and a corresponding target that we are interested in. The first step is to open up the double strand. This is done by adding the prime editor and the pegRNA. The pegRNA is a bit different from regular guide RNA, as it attaches to both strands, increasing the stability of the binding. The nickase activity of the prime editor causes a single-stranded break in the DNA, as dictated by the PAM site and the pegRNA. The pegRNA is designed to bind to the left part of the strand, and contains the template for the reverse transcriptase domain found in the prime editor. Next, the reverse transcriptase domain starts working, turning the pegRNA template into DNA. The new strand contains the edit but is otherwise identical to the original strand. Due to this, it will try to bind the same part of the unedited strand as the original one. The result is an equilibrium between the two flaps, as they compete for the unedited strand. Removal of the pegRNA and the subsequent ligation of DNA leaves us with an edited strand, indistinguishable from the original, except for the edit in our target.